Colorectal cancer is the third most common cancer diagnosis in men and women in the U.S. The risk for developing colon cancer is about 1 in 20, and although the number of deaths caused by colon cancer per year has been declining, there are about 50,000 people expected to die from it in 2018. In this video, we will be reviewing the mechanism of colon cancer, focusing on important key players. Given the mortality rates and the fact that most people are asymptomatic until very late stages, researchers have been trying to understand the molecular mechanism of the disease. It was found that 80 to 90% of colorectal tumors have mutations in a gene called adenomatous polyposis coli, or APC. The inactivation of both copies of this gene is often the first event in colon cancer. The APC gene on chromosome 5 encodes a 2,843 amino acid long protein with 8 functional subdomains. Most APC mutations that lead to cancer are those that remove the beta-catenin binding 20 amino acid repeats. On the left, we actually see the structure of a beta-catenin binding repeat from APC. APC plays important roles in cell cycle, motility, and adhesion. We will explore one of its most important functions in the VNT pathway. The VNT pathway regulates the stabilization of beta-catenin. Let's go through it step by step. When we have a functional APC protein, the VNT pathway is inactive. A destruction complex consisting of the APC, another protein called axin, and the enzyme glycogen synthase kinase 3, or GSK3, along with the enzyme casein kinase 1, or CK1, phosphorylates beta-catenin. Beta-catenin is phosphorylated at serine residues in its N-terminus and degraded. When beta-catenin is phosphorylated, there is no transcription of its target genes in the nucleus. On the other hand, when the APC is truncated and not functional, the VNT pathway will be activated. When active, VNT ligand binds to receptor complexes called Frizzled and LRP5/6. Then, a protein called disheveled, or DSH, is activated, and the destruction complex of axin, GSK3, and APC is inhibited. This causes the stabilization of beta-catenin, which then accumulates and travels from the cytoplasm to the nucleus. In the nucleus, it participates in the activation of genes involved in cell cycle, development, and cell fate determination. Understanding the molecular mechanism of a disease is essential for finding treatments. Researchers have been trying to find inhibitors of the VNT pathway. Pyruvinum is the drug that inhibits the VNT pathway by allosteric activation of the casein kinase 1, which is in the complex that leads to the degradation of beta-catenin. This graph shows the results of a cell proliferation assay called MTT, and pyruvinum reduces proliferation in multiple colon cancer cell lines. On the top, we also see that pyruvinum-treated mice show increased apoptosis compared to the vehicle control mice, as indicated by the tunnel assay. On the bottom, we can see that pyruvinum reduces nuclear localization of beta-catenin in APC mutant mice, and as we saw in the mechanism, less nuclear localization means less VNT signaling and more degradation of beta-catenin. JW55 is another small molecule inhibitor of the VNT pathway. It reduces tumor growth in APC mutant mice by stabilizing axin 2. A Western blot shows that colon cancer cells treated with JW55 have increased levels of axin protein compared to the DMSO control. We can also see decreased levels of beta-catenin with treatment. In addition to finding treatments, there has been an attempt to find easier and more accessible methods for detecting colon cancer. A new technique called digital protein truncation can detect mutations in fecal DNA from patients with early colorectal tumors. This technique amplifies APC genes using polymerase chain reaction and detects mutated or truncated proteins using in vitro transcription and translation of those PCR products. As seen in the big circle, only a small fraction of APC genes would be found in fecal samples. In this case, they sampled 2 to 4 molecules in each row, as indicated by the small circles labeled C through G. Then, protein products were separated by gel electrophoresis, and we can see the samples with mutant gene products. We can also see the results for actual patients using this technique, and the arrows and red circles indicate mutant APC products. In this video, we learned about the APC gene and protein, in addition to the VNC beta-catenin signaling pathways. Knowing the mechanism of a disease is very important for finding new treatments and detection methods. I hope you guys enjoyed this video.